So continuing on with part two of chapter 19, we've talked about the composition of blood, and now we're going to get into more detail into the erythrocytes, the red blood cells, the 44% of your blood volume. Okay? All right, so let's first go into the structure of the erythrocytes. You may have seen an image of erythrocytes before. Hopefully by now you in your career you have seen it. If you have not, this is the general structure. You see these this biconcave disc. They look like little circles. If they're healthy, and we're going to talk about if they're not healthy. I've mentioned sickle cell anemia already. If they're sickle celled, technically they are not healthy. That is not the shape that you would want. You want the shape that you see here, which we're going to talk about in more detail, why that shape is important and why we want to maintain that shape during the lifespan of the red blood cell. All right, so this is an image of erythrocytes, red blood cells. Remember that term. All right, so the shape and the structure in general and the components help the blood cell form and function. Remember, anatomy, form, physiology, function, very much related, right? So things are shaped a certain way for a reason. And the red blood cells, their shape is the shape that it is for a very specific reason. It's called you'll see the term erythrocyte, which we've already said. You'll see the term red blood cell, RBC, and you'll see the shape is classified as a biconcave disc, which is a sort of a circular shape, flattened disc that sort of has sunken in the middle, biconcave disc. What does this do? This increases surface area, which is very important because oxygen, different gases such as oxygen and carbon dioxide are going to flow across this surface, and you need to have increased surface area so that as much gas, oxygen as a gas, carbon dioxide as a gas, can flow across these surfaces as quickly as possible so that the red blood cell can function. Most mature red blood cells, as you see here, anucleate. It means once they become mature, they don't have a nucleus. And you may say, well, what is the significance of that? Why wouldn't red blood cells have a nucleus? They've been optimized for their specific function, right? So their function is going to be to transport substances, oxygen and carbon dioxide across the surface. And so once they've been optimized to transport, they've matured, and now they can do that. They no longer need a nucleus because they really don't need other organelles. You'll see in a couple slides that they don't live a super long time, so they don't need to continue to duplicate in that regard. So we'll talk about where they're made. We'll talk about how we continue to have new red blood cells. But one of the reasons that you can donate blood is this very reason. Red blood cells, your body makes more red blood cells. They don't necessarily need to duplicate themselves. Your body continues to make new red blood cells from bone marrow, which you've already talked about when we talked about the skeletal system, which we're going to talk about in more detail. So that plays a role into this as well. What we do need inside the red blood cell is hemoglobin. It's a protein, hemoglobin, which I hope you've all at least heard of, and that's the protein that's going to bind oxygen, right? So you don't have a nucleus taking up extra space. So that means you're not doing the things that the nucleus would do, which is help duplicate the cell, but you do have plenty of space for hemoglobin. So here we have the structure of the red blood cell, the erythrocyte, the RBC, as we have heard and called, right? So you have this biconcave disc, right? Biconcave, so two sides that are both concave. That means sort of dented in. That's what concave means. Concave, convex, concave, convex, right? So biconcave disc, and we see on the inside hemoglobin molecules. Oxygen binds to those hemoglobin. You have also cytosol and you have enzymes, right? The general structure of a red blood cell. Hemoglobin is large protein, consists of 4 polypeptide units, alpha chains, beta chains, bound to an iron-containing compound called a heme group, right? So the idea of alpha, beta chains, the idea of this hemoglobin is that you want to be able to bind oxygen ultimately. So an iron ion in each heme group is oxidized when it binds to oxygen in regions of high oxygen concentration, such as your lungs. Remember I mentioned before, your blood is pumped to your lungs, and that's where we're going to talk about that process. That's where the transfer between the oxygen we breathe in gets transferred to your lungs. The carbon dioxide that we're breathing out gets transferred from your bloodstream into the lungs. So oxygen comes in your lungs, transferred to the blood. Carbon dioxide gets picked up from your cells. It gets transferred to the lungs for you to eventually breathe out. Right, so this hemoglobin, which has these 4 polypeptide units, each polypeptide is bound to this heme group. The heme group oxidizes when it binds to oxygen and forms this red molecule called oxyhemoglobin. Red molecule. What color is blood? Red, right? Hopefully that starts to paint the picture. If you see oxygenated blood that has oxygen in it, it's red, a bright red actually. And so here is the structure, the erythrocyte structure. So you have the different heme groups, B chain, beta chain, alpha chain, these heme groups, which bind oxygen and become oxidized and form this, what we call oxyhemoglobin, which is a red molecule, okay? Oops. Hemoglobin, a little bit more information, releases oxygen into regions where oxygen concentration is low. I didn't stress that quite enough. So notice here, the iron in the heme group is oxidized when it binds to oxygen in regions of high concentration, high oxygen concentration, such as your lungs, right? So when there's a high concentration of oxygen, breathe in, you have a high concentration of oxygen in your lungs, your blood near the lungs will draw that oxygen into your bloodstream. At your cells, there's a low oxygen at your tissues and your cells in your body. When that blood's pumping through your body, your cells have a low concentration of oxygen. And so it goes from the high concentration in your blood to the low concentration in your cells. So it releases the oxygen when it's near your tissues that have a low concentration of oxygen. That's the general picture. There's more to the process, but that's the general idea of how oxygen gets from the outside environment into our lungs, into our blood, and then eventually disperse throughout our body. Now, this hemoglobin also binds carbon dioxide. This is not the only way that carbon dioxide is transferred in the blood, but the hemoglobin also can bind carbon dioxide where oxygen levels are low, and only accounts for 23% of the CO2 transported. It can also bind carbon monoxide. Don't get those confused. They are two different molecules. Carbon monoxide binds more strongly to the iron ion than oxygen, which is not good. That's why carbon monoxide is so poisonous, is because it can bind to the hemoglobin at a higher rate than even oxygen can. And therefore, if your hemoglobin is bound to carbon monoxide, it will not bind to oxygen as you want it to, and therefore your body eventually suffocates and you die. That's why carbon monoxide poisoning is considered very bad. They say it's an odorless gas, and so you can't smell carbon monoxide. That's why they recommend having things in your home that can detect levels of carbon monoxide. And if there are any issues, you can be notified and therefore make the necessary changes. All right, so here is the erythrocyte, the red blood cell structure with the heme group. All right. You don't need to be able to draw this diagram, just understand the concept. What is the lifespan of a red blood cell? And I mentioned this a little bit earlier. It's relatively short. Remember that they don't have a nucleus, so they're not making a whole bunch of new items once they have become mature. Right? Erythrocyte lifespan is only 100 to 120 days. Right? So they exist in this harsh environment. Remember, blood cells don't really stop moving. Your blood is continuously circulating, circulatory system. So there's continuously pumping and moving and bouncing against other blood cells and against the surfaces of your blood vessels, transferring oxygen in, carbon dioxide out, oxygen in, dioxide out, boom, 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 boom. So constantly working. So they're not really made to last a long time. They're existing for 100 to 120 days. Because they don't have extra organelles, they have primarily hemoglobin. They cannot repair themselves, and your body must continuously, therefore, make new red blood cells. How does that happen? Erythropoiesis, and some of these words are a little tricky to pronounce, and you may say, well, hey, I pronounced that a little bit differently, Professor. That's fine. Let's talk about it, and I want you to know the concept most importantly. Hematopoiesis takes place in the red bone marrow. Right? Formed elements in blood are produced by the hematopoietic stem cells. Right? Specifically, erythropoiesis, and you will see this term probably on a quiz or exam. You will see this term. It's the process of making erythrocytes. Right? It takes about 5 to 7 days. Not very long. So if red blood cells are only living about 100 plus days, we have to continue to make new ones. It makes sense that it only takes about 5 to 7 days to make some new ones, and the whole process is sort of detailed here. What I want you to know is that this takes place in the red bone marrow by the hematopoietic stem cells, and that it takes about 5 to 7 days for it to happen. And then here on the slide, it goes through the process, and we don't you don't need to understand the entire process of differentiation, erythroblasts. You don't need to understand every step in this process, but I do like this. I do want you to point out, as we mentioned before, right here in this step, the nucleus in the erythroblast shrinks as it matures, eventually ejected, resulting in reticulates into the bloodstream after ejecting remaining organelles. So remember, so these red blood cells originally have organelles, originally have a nucleus, but those get ejected by exiting through the pores and the sinusoidal capillaries of bone marrow. This is the diagram just showing you that these stem cells form these erythrocyte cells that are going to be erythrocytes, and this is in the red bone marrow. This is why bone cancer can be very dangerous, because you need red bone marrow to make red blood cells. So you have this early erythroblast, the nucleus is then ejected, the remaining organelles are ejected, and the cell enters the bloodstream and it forms what is a mature erythrocyte, which has been optimized for gas exchange. Right. You can pause this video at any time if you feel like I'm moving too fast, and you can actually speed it up if I'm moving too slow. What regulates erythropoiesis? It's a negative feedback loop. Hopefully that rings a bell. We've talked about negative feedback loops in Anatomy and Physiology Part 1. Right? So it maintains the hematocrit within a normal range. Remember hematocrit, remember that term, that your red blood cell count. Right, so if your blood levels of oxygen fall below normal, okay, so your body constantly monitoring itself, and if the blood levels of oxygen, right, so your body is noticing that you have less oxygen in your blood, if nothing else has gone wrong, it just means that there are less red blood cells. Remember, red blood cells only live about 100 to 120 days, so blood cells have died. Therefore, there are less blood cells to carry oxygen, so your body notices that there's less oxygen in your blood. Kidney cells detect the falling oxygen levels. We're going to talk about, when we talk about the urinary system, we're going to talk about how all of your blood goes through your kidneys. That's how your blood is flushed out. Now, oxygen comes in your blood at your lungs. Carbon dioxide leaves your blood at your lungs. But in your bloodstream, you also have, we're going to talk about, this is not carried inside of the red blood cells, but carried in another portion of the bloodstream is going to be waste products, which we haven't really gone into details about yet. So, but to get those waste products out, you don't breathe them out like we breathe out carbon dioxide. It comes out as urine. So we'll talk about that. So just keep that in mind. All of these things play together: respiratory system, cardiovascular system, urinary system. And we'll even talk about the digestive system, transporting nutrients through your bloodstream. These all work together. And remember, all these, even from Bio 109, all of these body systems are working together. So you want to sort of see that big picture, right? So blood oxygen levels drop, which is detected by your kidneys. Kidneys produce more erythropoietin and release that hormone into your bloodstream. So the kidneys release the hormone. So a negative action happens. Your oxygen levels drop, which is noticed by your kidneys. They release the hormone erythropoietin. That hormone causes of the erythrocytes to increase in your red bone marrow, and eventually more blood cells are created 5 to 7 days, more blood cells are created. Those more blood cells can carry more oxygen. You have more blood cells to carry more oxygen, therefore the oxygen levels in your body go up. We don't need to make as many red blood cells right now. All right, that makes sense. And so that's a diagram just showing you that whole process. You can look at that slide and hopefully understand it. Erythrocyte death. All right, so we have erythrocytes can become trapped in your spleen, and we'll talk about the role of the spleen, the role that it plays in this whole process. But in general, erythrocytes can become trapped in the sinusoids of the spleen. The spleen is an important organ because of this. Spleen macrophages, which you're gonna see this term macrophage often, especially when we talk about your body's immune response. But macrophages digest erythrocytes. Hemoglobin is broken down into amino acids, iron, heme. Heme is first converted to waste product biliverdin, and then can convert it further into bilirubin. Ions, and amino acids are recycled, used to make new hemoglobin. Your body recycles. Ions are transported back to the red bone marrow in the bloodstream. And bilirubin is sent to liver for excretion. All right, so that's the only part that's excreted. Right, so from the heme, we get the bilirubin, which is then eventually wasted. That's the only product that's really wasted and excreted. So these red blood cells, which we know live 100 to 120 days, go to the spleen basically to die. But parts of them do get recycled. The iron and the amino acids do get recycled. Remember, amino acids make up proteins. Remember, hemoglobin is a protein, all right? And then, but the actual heme group is converted to a waste product, bilirubin, which is sent to the liver for excretion. Right? So that's the process of erythrocyte death. And it's just showing you that diagram here. You have this slide, hopefully you can look at it in more detail, really study it, study this process. I have seen a question asked on this bilirubin on a previous departmental final exam. So you made this line heme converted to the biliverdin and then eventually to the bilirubin. You may want to make a note of that, and the fact that the iron and the amino acid are recycled. So even though I haven't yellowed those, you may want to make a note of that because I have seen those mentioned on the final exams. But you know, those can change. Okay. Anemia. I've mentioned one form of anemia, sickle cell anemia, but that's just one type of anemia. There are other types. Anemia is defined as decreased oxygen carrying capacity of blood, right? So we talked about the kidneys, recognizing that there's less oxygen in the blood, right? We mentioned that. Anemia in general, which can have different causes. There can be, you know, death of blood cells. There can be blood cells that have an incorrect shape. But in general, the oxygen carrying capacity of the blood has changed when it comes to when we talk about the term anemia. So, you know, a couple blood cells dying to the point to where your body starts to reproduce new blood cells is not really considered anemia, right? It's actually the red blood cells not being able to carry oxygen, right? So 3 primary causes. Sorry, I was listening to my children for a second. 3 primary causes: decreased hemoglobin, Remember, hemoglobin binds oxygen. Decreased hematocrit, which means we have a lower number of blood cells. Abnormal hemoglobin, right? So all of these can cause problems with the oxygen carrying capacity of blood. General symptoms include pale skin. You don't have enough oxygen in your skin, it's going to be pale. Fatigue, if the body doesn't have oxygen, then it can't function properly. You need oxygen to be able to, for your body to make energy. So if you don't have oxygen, you're going to have less energy. So you're going to be fatigued, you're going to feel weak. Shortness of breath. Obviously, if your body can't hold out, if it can't transport oxygen, you can have trouble breathing. So this can be caused by many different different types. There can be different types of anemia. Severe anemia can elevate the heart rate. Your heart can try to compensate because you're not able to. Your blood is not carrying enough oxygen. Your heart will try to pump harder so that more blood circulates back to the lungs, and so has to pick up, the blood comes back to pick up oxygen more often. And so now that blood is pumping faster through your body. So your heart rate increases to try to match this, the demand for oxygen in your tissues with the lack of oxygen that's being carried by your bloodstream. It can cause all types of issues and problems as you can imagine. So anytime where our bodies have a lack of oxygen, there's going to be a problem. Very simply put. One of the most common forms is iron deficiency. If you've ever given blood, one of the first things they do is they check your iron at the Red Cross. I try to donate blood. One of the reasons being simply because I know my wife suffers from sickle cell anemia and has had life-saving blood transfusions. And we can talk about blood transfusions. Those are actually very interesting science, to be able to take somebody else's blood and put it into another person's body. How can we do that? And that person, live? That's amazing science right there. You may think of it, people have all been doing it for years, so you may think of it as something simple, but it actually is quite complex. But if you have low iron, they won't let you give blood. Iron helps make up that hemoglobin, which, if having low iron, can be due to inadequate nutrition, or your body just doesn't absorb iron. And without this, without iron, your heme group won't function properly. Your erythroblasts will be unable to synthesize hemoglobin, which then your body won't be able to bind to oxygen. The hemoglobin, your red blood cells won't have the hemoglobin necessarily to bind to the oxygen, right? So that could be an issue. Reduced iron could lead to reduced hemoglobin, which leads to reduced amounts of oxygen in your body. Decreased hematocrit. You can blood loss, and we're not talking about just normal blood cells dying. We're talking about blood lost. I mentioned if you get shot, if somebody were to get shot or stabbed, I know that sounds terrible, but it's true. It happens every day, and one of you all in this class may be working in that area one day. If you lose a lot of blood, therefore you have what we classify as anemia, significant loss of circulating erythrocytes, and that reduces your blood's ability to be able to transport oxygen. Therefore, you have anemia. And so that's another form of anemia that can happen. Infections can cause anemia, erythrocyte destruction, as you see on the third point here. you can have, so an infection can cause an unusual amount of red blood cells to die. Certain medications can stop the production of red blood cells, which could cause anemia. It's called aplastic anemia. And then I've mentioned sickle cell anemia before. Sickle cell is an abnormal hemoglobin. It's abnormal shaped. Remember, hemoglobin is what makes up the majority of the inside of a red blood cell. And you have these sickle-shaped cells which have an abnormal hemoglobin in them. And people who have sickle cell, not all of their red blood cells are sickled, or they wouldn't be able to transport any oxygen, but it usually a percentage of their cells are sickled, and therefore their red blood cells transport less oxygen. Their red blood transports less oxygen overall versus a healthy individual. But the sickle-celled red blood cells don't bind as well to oxygen, right? So that's the issue there with those with sickle cell. And this is just just showing you the hemoglobin S molecule, which causes that causes these cells to sickle shape, and it leads to the erythrocytes destruction, reduced amount of red blood cells, which causes a reduced amount of oxygen being transported by the blood. We're going to talk about the white blood cells, WBCs, which are leukocytes, and the immune function. All right, so this is another part of blood. Remember, we talked about plasma being 55% red blood cells, 45%, and then another 1% of other molecules that are in blood. Okay, so remember that. So we've already talked about the red blood cells, which play the huge role in transporting oxygen and carbon dioxide. They live about 100 to 120 days. They take about 5 to 7 days to be made. They're made in the red bone marrow. If you have low oxygen or low oxygen levels in your blood, that is considered an anemia. That can be, hemoglobin that has the wrong shape, sickle cell anemia. It could be a loss of red blood cells, big blood loss, or due to an injury. It can be blood loss due to disease, or blood loss due to your body's ability not to make new red blood cells due to some type of medication you're taking. So these are all things you want to remember as we move forward. Leukocytes are your white blood cells. Leukocytes, as we saw in that image in section 1 of this chapter, are larger than your red blood cells. They do have a nucleus. Their nucleus has not been ejected, so they do have a nucleus. So that means what? They'll probably live a little bit longer than red blood cells, but they use bloodstream as methods of transportation. They are transported primarily through your blood, but generally don't perform their functions within blood. What does that mean, Professor? So these are more primarily for an immune response. So let's say I get unfortunately infected with a virus. That virus is not necessarily infecting cells in my blood. It's not necessarily infecting my red blood cells, but my blood travels all throughout my body. And so let's say that virus has infected a certain cell. My blood cells my white blood cells can travel through my blood, my bloodstream, and the blood vessels, to be able to eventually get to wherever the infection is. Okay, so they generally don't perform their functions within the blood, but they use the blood to travel. Your white blood cells, your leukocytes. All right, so remember, erythrocyte, red blood cell, RBC, leukocyte, white blood cell, WBC, right? Hopefully you've read these in a chapter and so they're already somewhat familiar to you. Okay. Leukocytes are divided into two basic categories, which don't seem immediately obvious, but these are the categories that we use. Granulocytes, which contain these cytoplasmic granules that are released when activated, and you have these agranulocytes, which do not have visible granules. And so you'll see this. Granulocytes, readily distinguished by their unusual nuclei, single nucleus composed of multiple connected lobes. I think it's best to, when you see the image, you'll see the difference between granulocytes and agranulocytes. But cells in this group contain general lysosomal granules. Right? And these are divided into three categories based on the color of the granules, which can have different colors. Then you have, which can have different colors when stained with basic dyes. Okay, so the granulocyte category of leukocytes. Let's go into these. Neutrophils. Most common leukocyte, they have cytoplasmic granules that have a certain color to them when they're stained with the dye. That's how we distinguish it. I know you're saying these are white blood cells, the immune response. Why does the color of the cell, the color of the granules in the cell, but that's how we distinguish different leukocytes, as I said. Doesn't seem like the most ideal way to distinguish it, but that is the way we do it in science. And I highlighted usually a key point for each of these different leukocytes. All right, so we have neutrophils, most common leukocyte. Remember that it is a it is a granulocyte. So it's a leukocyte, a white blood cell, most common granulocyte, known as neutrophils. Active phagocytes that ingest and destroy bacterial cells. Phagocyte, ingest other objects. Phagocyte by phagocytosis. So they can ingest and destroy bacterial cells. Injured cells release chemicals that attract neutrophils. That's called chemotaxis. We'll talk about that process in more detail a little bit later on. They exit the bloodstream. These neutrophils can then exit the bloodstream and release granules in damaged tissue. So, bacterial infections, they directly work against those. Eosinophils. Eosinophils appear red, and they appear to be red due to take-up of eosin dye. These phagocytes, neutrophils, lilac color, eosinophils, red color. Phagocytes that ingest foreign molecules, right? Phagocytosis brings objects into the cell, then destroys them. Remember that lysosome term? Remember granulocytes, all cells in this group contain general lysosomal. Remember lysosomes from when you studied that in chapter 4, I believe, and Bio 109, when you talked about the cells and the different organelles of the cell, lysosomes, right? Lysosomes can destroy certain objects in the cell. So once these neutrophils or these eosinophils absorb these bacteria or foreign objects, depending on which one it is, then the lysosomes can work to digest it, destroy it, basically. Okay, so that's just knowing this is how our immune system works, and we're going to go into more detail about the immune system later on. But so we have neutrophils, lilac color, most common granulocyte, most common leukocyte, and we have the eosinophils, which are going to digest foreign molecules, respond to infections, parasitic worms, allergic reactions. Granules contain enzymes and toxins specific to parasites. They also have chemicals that mediate inflammation. Inflammation is a huge topic, which we will discuss. Basophils, least common leukocyte. So you have neutrophils, most common, basophil, least common. These are all granulocytes, right? Granulocytes, remember, single nucleus composed of multiple connected lobes, and they have a specific color to those cytoplasmic granules, okay? All right, remember those terms because we will see those, these are important. Basophils appear to be dark purple. Dark purple color, chemicals and granules mediate inflammation. Notice the eosinophils also mediate inflammation. Basophils, that's one of their most important functions, is that they mediate inflammation. All right, so lilac color, neutrophil. Red color in the eosinophil, and then dark purple in your basophils. I know they look similar, but they are different. And then we have agranulocytes, which means a lack of granulocytes. Include lymphocytes and monocytes. Both lack cytoplasmic granules, but do contain lysosomes. Right? So lymphocytes, second most common leukocyte. Most common leukocyte is your neutrophil, right? Second most common is your lymphocyte. Two basic types in appearances but different functions. Both types are activated by cellular markers found in all cells called antigens. So you have two basic types. You have B lymphocytes, when activated, produce antibodies. That's going to be very important when we talk about your the different levels of your immune response. We'll talk about antibodies. When activated, produce antibodies which bind to and remove antigens from tissues. Each population of B lymphocytes secretes antibodies that bind only to specific unique antigens. And why is that important? We'll talk about that. You also have T lymphocytes. Hold on, move my activated by specific antigens, but do not produce antibodies. These T lymphocytes activate other immune system components that directly destroy abnormal body cells. T lymphocytes. So remember the B and the T lymphocytes, which we short-term call them B cells and T cells. These are agranulocytes, which are white blood cells, leukocytes, agranulocytes, and they're both in the lymphocyte category. Right, and then we also have monocytes, which is the largest leukocyte. Right, so neutrophil most common. The B cell, T cells, second most common. The monocytes, largest. Basophils, least common. I hope I got those right. But you can see those in the slide. But just know that that general idea. All right, so this is the largest leukocyte. Only circulating blood briefly before exiting capillaries to inner tissues, where some mature into macrophages. We've talked about macrophages a little bit already. These are phagocytic. Again, phagocytic cells that ingest dead and dying cells. Remember in the spleen, we talked about macrophages ingesting red cells go to die. So ingest dead and dying cells, bacteria, antigens, and other cellular debris. And they activate other components of the immune system. All right, we're gonna see why that's so very important. All right, so we have lymphocytes, and the lymphocyte, which is your B and your T cells. We also have the monocyte. Okay, that's the different structures there. Complete blood count, important test for anemia and other conditions. Right, knowing, we talked about the hematocrit, knowing the amount of red blood cells, but a complete blood count is also something that would be very important. Red blood cell count in cells per milliliter is used to calculate your hematocrit, right? How many red blood cells you have, but you also want to know your hemoglobin concentration. All right? We can know how many red blood cells we have, but we need to know how much hemoglobin we have as well. You want to know the characteristics of your red blood cells, the size, the volume, concentration of hemoglobin in the cytosol. Remember, you can have red blood cells and they be sickle-shaped, which are not going to be very effective. So just because you have red blood cells doesn't mean you have adequate ability to be able to carry oxygen. Platelet count is going to be very important, and volume. That's another thing we can, when we get a complete blood count, we want to know how many platelets we have. So that's our ability for our bodies to be able to heal. And then we also want to know the numbers and types of leukocytes, especially when you have certain treatments. You can have a reduction in the amount of leukocytes, which means a reduction in your ability to be able to ward off disease. That would be an issue. If your white blood cell count goes down, you're more susceptible to illness, infections, and getting sick, and letting that sickness propagating throughout your body. Therefore, that could be a major issue. All right, let's talk about leukopoiesis. Right, we talked about the formation of new red blood cells. We also need to talk about the formation of new white blood cells, WBCs. So, as I said before, with the red blood cells, you don't need to know the entire process of how these cells are formed. They start off with the myeloid cell line, which produces most formed elements, including erythrocytes and platelets. They also have this lymphoid cell line, which produces lymphoblasts, which eventually become your B and your T cells. Right, so different types of different cell lines lead to different types of cells. And this talks about how the stem cells differentiate into different precursor cells. I want you just to read through this, but like I said, you don't need to know the whole process. The B and T lymphocytes mature in different locations. B is for bone marrow. Remember, both of these B and T lymphocytes are agranulocytes. B cells mature in the bone marrow. T cells mature in the thymus, which plays a role into how they eventually will circulate in our bodies. And this is a diagram just showing you the overall structure of how these blood cells these white blood cells are made. And you can look at this slide in closer detail and just see where these different cells come from in our body. Leukemia. You may have not put those two things together, at one point. Leukocyte, leukemia. Maybe you didn't think those two terms were related if you had heard both of those terms. But leukemia is cancer of the blood cells or bone marrow. Right? Acute rapid increase in immature, non-functional, poorly functional blood cells, which can be problematic, and then chronic, slow accumulation of abnormal, mature leukocytes, white blood cells. Right? That can lead to what we call leukemia. Right? So different forms of leukemia. Like I said, it's cancer of the blood cells or the bone marrow, blood cancer, basically. Can be very dangerous. Abnormal cells can crowd your marrow, reduce your ability to manufacture healthy cells. Cancerous cells can enter the bloodstream, and remember, your bloodstream is connected throughout your whole body. So now those cancerous cells can spread to other parts of your body and cause cancer to grow, metastasize in other parts of your body. So it can be very, very dangerous to have